Please take a seat. It is, let's say, a great pleasure for me to introduce Paolo Casperini from the University of Trieste in Italy. And I think he has a lot of experience in advanced therapy and in the role of the EMA committee, which helps us to bring really in a hopefully fast track such kind of innovative therapies, at least to the proof of concept trials. Another important point is, of course, that we have to have the vision to bring it really on the market, because that is what we need. But firstly, it would be nice if we have a good support by regulatory authorities to bring it in the proof of concept phase 1 to A to B trials. Thank you for being with us.
Thank you, Chair, and thank you to the organizer for inviting me. I will give you a very brief overview on the role of the Committee for Advanced Therapy of the European Medicine Agency and the work we are doing month by month in evaluating ATMPs. First of all, what are ATMPs? From a regulatory point of view, belong to this category, either gene therapy, medicinal products, as well as somatic cell therapy, medicinal products, and tissue engineered products. In all cases, the development requires high specialization and know-how, and product safety and efficacy profile must be carefully, prospectively planned in order to produce robust and reliable data. There are some quite common features, risks, and limitations to all the three categories of ATMPs. There is an intrinsic variability and possible microbial contamination of starting material, tumorigenicity, dedifferentiation, cell loss of function, immunogenicity, ectopic engraftments of cells to non-target tissues, shedding, germline environment, limited production and thus quantity limits for sampling and testing, very small batches, reduced shelf life, and patient may need to be administered with out of specifications products, as currently they are facing with some of the ATMPs we already approved for the market. So the intrinsic nature of ATMPs requires a regulatory flexibility. This is very clear, and for this reason, within the EU legislation, it has been included what is called risk-based approach, RBA. And so the risk analysis should comprehend the whole development in order to determine the amount of data needed for marketing authorization. And there are, of course, existing lines for this risk-based approach. And the role of the EMA is to classify ATMPs, then to give scientific opinion, scientific advices on ATMPs, and also at the end to the assessment of marketing authorization of ATMPs. And the members of the committees, and there is one delegate for each European country, plus some representative of patients and physicians associations, is to evaluate the quality, the non-clinical, and the clinical aspects of each ATMPs. At the end, the goal is to safeguard public health through regulation and evaluation of medicinal products, and treatment safety also via stringent, eventually post-marketing authorizations. The CAT is here, interacts with a series of different committees already working at the European, at the EMA. And in particular, from the Scientific Advice Working Party, then a very strict link between the CAT and the CHMP, because these two committees are those who committee which at the end are in charge for the final marketing authorization. And usually we start from this first question, trying to answer, is the product an ATMP? So there is a classical ATMP classification procedure. If yes, could the product be used in children? If yes, of course, there should be a pediatric investigation plan, or PIP. Is the product for a rare disease? There, it can have access to what is called the orphan drug designation. So it starts the procedure for orphan drug designation. And is the product for a priority medicine or for an urgent unmet need? So, and in this case, there should be the possibility of an accelerated assessment. So if at the end there will be sufficient data and the risk-based approach and the results of risk-based approach are positive, then the ATMP can get the standard marketing authorization with or without obligation to conduct post-marketing safety efficacy study. If not, could be rejected, or in some specific cases, like for example, emergency situation, seriously debilitating life-threatening condition, orphan, unmet medical need, benefit risk positive. At the end, there could be a conditional marketing authorization or a marketing authorization under exceptional circumstances. During this last few years, we have been involved in approving a series of ATMPs. You know most of them. The most interesting one, in my opinion, is Glybera, because it was the first gene therapy product approved on the market for in EU. And it was really, I was co-op of this product. It was really a fight. I'm not talking about the quality of these ATMPs, but I'm talking of the relevance of this, of the work we did as the first product. So we were able to open the door to for the whole following ATMPs that have been then after that approved on the market. Also extremely relevant was the approval of Holoclar, which is the first cell therapy approved at the European market. And then a series of additional ones, including some which have CAR-T already presented, discussed during these 2 days of meeting here. Yescarta and Kymriah in 2010, Luxturna, the first gene therapy for retinitis pigmentosa, always in 2010, and finally in 2019, Zynteglo, the gene therapy for beta thalassemia. Some comments on this first experience, and the comments that I would like to share with you. I am a physician and I'm a scientist. Within the committee, there is a mixture of people coming directly from the regulatory field and with a direct link with the national agency, and some minority of physicians and scientists. So the first, there is still a need to define a common understandable alphabet among all of us. Just to give you an idea, according to the EU legislation, red blood cells are not cells because they don't have the nucleus. And of course, platelets are not cells. I am an hematologist, apart from also being a medical geneticist. If one of my students comes to during the examination saying the red blood cells are not cells, he or she will never pass the examination. But this is the legislation. It's not an issue of the cat, but this is the current existing legislation in EU. And then still we have discussion on substantial manipulation, because what is substantial for me could not be substantial for someone else. In the past, there were a long discussion on the definition of living organism or living cells, and also on human gene therapy. You should consider that according again to the EU legislation, if you are genetically modifying a bacteria like a salmonella, and this bacteria pass through the bowel and release in the bowel a genetically modified protein, and then it is eliminated through the feces, this is considered human gene therapy. So it's quite far away what we are considering as human gene therapy. But again, this is the EU legislation. So, and then indications. And again, it was a very strong debate and fight to completely change the indication. So far, the indications were mainly based on signs and symptoms. But if you consider that for many rare diseases, genetic diseases, the clinical classification are very heterogeneous, and in some cases defined years ago, in some cases defined even decades ago. So at the end of a long discussion, we were able to, at the end, include in the indication the specificity of the presence of B allelic mutation, in this case is Luxturna, within one specific gene. So it's completely changed education. The education is very broad. I think, if I'm not wrong, it's like visual loss. So something very, very general without any specific subclinical entity. But due to B allelic mutations in RP65 gene. This is something which for us is very, it's currently daily based work, but was not that easy to get approved by the other colleagues, either in the committees, in the CAT, as well as in the other committees. And then what we learned, for example, again, thinking to Luxturna, this is something I would like to tell to the companies. In many cases, we were talking about science symptoms of the disease, but science symptoms for people who never has been involved directly in evaluating a patient, or who they not have any clear clue about the disease, are signs and symptoms. So visual loss is visual loss. But if you are able, because if in the case of Luxturna was possible, to come to the attention of the committee with videos showing the impact, the dramatic impact in terms of efficacy and benefit for the patients, like in the case of Luxturna, people not able to even walk and jump over obstacles in a dark room before the treatment, and the ability to perform those tests after the treatment, that was just fantastic. After the video, the attitude of people mainly coming from the regulatory field completely changed towards the considering a very useful and beneficial treatment. And then another big issue is how we should rethink to statistical approaches. Usually we have very low number, and so the classical and conventional statistical approaches should be completely reevaluated. So, few words regarding genome editing. We start discussing genome editing also not only because it's extremely relevant, but also because we have been asked to give scientific advices on some genome editing protocols. I'm not going into detail because you are much more expert than me in genome editing, but this is the major issue we have been discussing, and of course is the issue of off-targets. For two reasons. First of all, because there are no gold standard methods to evaluate and quantify the off-targets. And in the regulatory field, people would like to have standard methods and reproducible standard methods to evaluate the products. And the second, of course, there is the discussion on variable of unknown significance, the need to consider tissue specificity. So the interpretation of off-targets remain a challenge. What current CAT, which is the current position of CAT, is that all genome editing technology in medicine should undergo the same regulatory scrutiny by CAT as ATMPs. The risk-based approach should be applied to genome editing. Still, we have to discuss about the current regulatory and clinical trial frameworks, and then I will tell you why. Of course, there are implications on immunogenicity. There are issues related to genome editing products and the GMO regulation. Quality requirements on gene editing, starting materials, promote state-of-the-art GMP facilities, and relevant and necessary non-clinical requirements. And as regard the orphan drug designation, which is, whose goal is to promote research on treatments for rare diseases, we consider that genome editing is compatible with these rules, but additional work should be done to better define the whole scenario. In conclusion, regarding the genome editing at CAT, we consider this technology really promising, but right now, only with limited clinical trials developments worldwide so far, but challenges in developing these products. And what we are planning to do and we're doing is to continue to listen to developers, support development by fostering early regulatory dialogue and cooperation to facilitate marketing authorization. And what we did or doing, we published in September 2008, 18 a report on EMA expert meeting on genome editing technologies used on medicinal product development, a meeting we organized at EMA. Revision of the CAT-CMPG guideline on genetically modified cells, reflection on other guidance, harmonize environment and GMO requirements. So in conclusion, I think we should consider a series of currently existing rules and regulations in this field because they are extremely relevant even for you in developing the products and having at the end the products approved for the market. The first is market exclusivity, 10 years. So protection from similar products only, but it can breach it by a second similar product only if it's safer, more effective, or otherwise clinically superior. This is something very difficult to demonstrate in practice. Let me say that in my opinion, it's almost impossible to demonstrate based only on clinical superiority. So then there is the issue of non-similarity, which was put in place to protect, but products for same indications can access the market. Similarity is a big issue. Some regulators consider similarity by function, which make extremely difficult to define a new product non-similar if the therapeutical target and the category are the same. Consider gene therapy, or even more, start considering genome editing, in which you are targeting different mutations within the same gene. So, and in principle, they can be considered different protocols or considered the same by function. Then another issue is that in many cases, the number of patients is very low. I'm talking mainly regarding rare diseases, making it difficult to design clinical trials with a new ATMP because it's impossible to reach acceptable numbers. And this is a very general rule. But this is further hampered by the fact that if an already approved ATMP exists with positive safety and efficacy data is unethical to not offer it to a patient and ask him or her to enter a clinical trial with a new product, even if it's more promising. And in the end, what about unmet medical need? If something already exists, able to treat, how is possible to overcome this relevant issue, which is the presence of unmet medical need to develop and to get approved one ATMP. So at the end, the risk is to stop the development of new ATMPs for rare diseases for which an ATMP is already existing. And the second group of consideration is with increasing use of genome editing and the following deeper personalization of products. So custom therapy for most of inherited diseases, several scenarios might arise. The first one is the extensive use of hospital exemption. The second is difficulties in carrying out clinical trials. Or, as I said before, if you are targeting different mutations within the same gene, but then you have different oligos, how can you consider them? Different products or the same products? And in any case, how can you compare the data in a clinical trial? And of course, there is a need to rethink to similarity and to orphan drug designation. Let me say a few words about the follow-up and registries. In principle, should be good to have long-term follow-up and the registry, but with ATMPs, and keep in mind that the registries are extremely expensive to have and develop and maintain. And ATMPs are mainly based on rapidly evolving technology. So the information you can collect will be simply useful to increase our general knowledge, but not to impact on each specific product, because no single ATMPs currently in use will be in use in 5-10 years from now. We are almost sure about this. And finally, the personalization of therapy is based on omics data. We have been asked to evaluate a series of new products, especially immunotherapy for cancer, in which the product is based on genomics information, genome sequence information. So open a series of issues on the ownership of the data, access and use those data, especially if the whole process is managed by a company. And this last slide is to thank you for the attention and to invite you to come to Trieste next summer. Trieste will be the European City of Science ESOF in 2020. Thank you.
So this paper is, first I have to thank for this overview and the strategy of the EMA. Secondly, this paper is now open for discussion. I hope that many questions rise. No any? I'm surprised. Because let's say, I have a few. One point is what you really think about the substantial modification of a product, because if you start to develop something, and this is what we are doing here and pushing forward, then you have to be aware of that you have to modify at the end your product based on, let's say, the experience you get from the patient, because whatever you are doing with a risk assessment, you cannot prognosticate, let's say, any kind of outcomes or of problems and so on, which means you have to modify your product if it is useful. But then you have to start again and again and again. And this is a little bit difficult if you are in competition to other agencies like the FDA, who has a more liberal view, in particular if we deal with first-in-human phase 1 to 8 trials. What is your comment?
What we discussed a lot, as I said before, is the definition of substantial manipulation. And of course, there are some different views. There is a kind of general agreement which substantial manipulation is considered like a series of centrifugation at the very early beginning of the procedure. If you collect, for example, blood cells, and then you have to manipulate centrifuging cells, and then increasing or decreasing the temperature, and add something like, I don't know, chemical compound that at the end is considered substantial manipulation. As regards the other possible changes during the process of marketing authorization, this is something we are experiencing, for example, with Zynteglo, in which there was a process, it was a change in manufacturing process. And so far, we discussed it with the company, and at the end, we reached an agreement in order to don't stop having Zynteglo in the market, but demonstrating enough pressure on the company, try to get back to the first manufacturing process, which was the one we evaluated, and at the end was the really good one as compared to the second one that is slightly changed. Okay. But you are right. I mean, the legislation in between FDA and EMA are a little bit different. I don't know which is the best one, to be honest.
Sure, nobody knows, but it makes something easier in the first phase of the development to be more flexible. Yeah.
This is something we are trying to do. And as I said to you before, I am one of the few physician and scientists sitting in the committee, and with the other colleagues, at the end, we were able to convince people coming from the regulatory fields that, at the end, it's better to have one product. Once the safety is not, and I mean the quality and the safety issue are solved, that at the end it's a good option to have on the market alternatives for the physician to treat patients. Okay. If at least a little minimum benefit. And now the situation is changed. The attitude of CAT, also with the PRIME, which is a accelerated procedure, is more flexible, and they are much more open-minded in evaluating all the ATMPs.
Okay, yeah, I see a question. Wonderful.
Good morning, Michela Gabaldo from Teleton Foundation. I would like to ask you to elaborate a little more on the last comment that you were saying on the fact that in the next 5 to 10 years, the current ATMP will not be more used. Could you elaborate a little more? Just because it is arriving the next generation, or because, I would like just to have your thought on this.
Yeah, I mean, it's a kind of very rapidly evolving technology, first of all. And so there are clearly differences and improvements either in the technology per se, changing of viral vector, for example, as well as the use of, for example, genome editing. And if you think what we approved in 2012 or even 2010 and 12, they are not anymore on the market, but there are new ones. So it's the overall number of ATMPs we are evaluating or we are giving scientific advice is increasing month by month. So it's almost clear that it will be very difficult to have patients treated now, which will be treated for a given disease, and additional patients treated with the same product in 5 years, 6 years, even from now. The technology is really rapidly evolving. Even CAR-T, we are evaluating new CAR-T. And I'm not saying that they will be approved on the market, but are a kind, as we listen also from the previous presentation, a new generation of CAR-T. So most likely, I don't know which will be the lifespan of Yescarta or Kymriah or additional ones that are in the pipeline of being approved, but most likely will be, I don't know, 4 or 5 years.
Okay, no further questions so far. Then we move on, and thank you, Paolo.
Thank you, Paolo. Okay, our next speaker is Timothy O'Brien from the National University of Ireland. He will talk about MSCs as therapy for diabetic complications. So welcome.
Yeah. Thanks. Thank you very much, and thanks for the opportunity to participate in the meeting. I'm going to present our experience probably over the last 10 years or so in bringing 3 preclinical ideas forward into clinical trials. And I'm presenting it from the context of an academic center engaging with partners across Europe. And I thought I would do it and contemplate some of the challenges we've had in traversing this terrain, maybe in the context of what an entity like Restore could help with. I'm an academic clinician, so I work half-time in the clinic, half-time in the university, but I'm also a founder, director, and equity holder in a company called Orbsen Therapeutics. So I'm a diabetologist, and my clinical practice is mainly focused on caring for patients with diabetes. And this just global map shows the pandemic, which is before us, with a predicted 552 million affected by 2030. Diabetes is complicated by multiple vasculopathies, which can be considered macrovascular or microvascular. And I'll talk about a translational path in macrovascular disease, similar to what we heard from the last talk in the first session this morning on critical limb ischemia, and then in microvascular complications, focusing on nephropathy and non-healing neuropathic ulcers. So the focus of our work is the bone marrow-derived mesenchymal stem cell, and all of the work I'll present during this talk will focus on that cell, the bone marrow-derived mesenchymal stromal cell proliferating on plastic. And we're interested in the use of MSCs for diabetic complications, more from its paracrine actions and immune reprogramming actions, rather than its capacity to differentiate the bone, cartilage, or adipose tissue. So when one is considering MSCs as a potential source for these therapeutic paradigms, one considers the source, adipose, bone marrow, umbilical cord. Whether the cells are bulk manufactured or sorted, whether one uses an autologous or an allogeneic approach, whether one uses flasks or more recently available bioreactors, and the avoidance of fetal bovine serum in terms of xeno-free approaches. So critical limb ischemia, already alluded to earlier today, a condition of unmet medical need, with about a third of patients having no option for revascularization and therefore requiring amputation. So many groups have seen that these mesenchymal stromal cells from both non-diabetic and diabetics secrete multiple angiogenic factors. When one takes the conditioned media from mesenchymal stromal cells and applies it to tubulogenesis assays, there's an enhancement of tubulogenesis. And in this preclinical in vivo model, when one takes bone marrow MSCs, either from a diabetic source or a non-diabetic source, and transplants it into an acutely ischemic hind limb, there's an improvement in vascular recovery. And when we think of preclinical models, I mean, this rodent acute ischemia model is the most commonly used model in progressing through the translational pathway, starting with animal efficacy, but yet it doesn't really reflect the disease in humans, which is chronic and associated with multiple risk factors over many years. There was also an improvement in the animal's ability to ambulate. So this preclinical dossier, also on this slide, you can see the ability of the cells to promote muscular regeneration. So these animals have been injected with bone marrow MSCs, and the one in the upper panel have been injected with saline alone. So we then move on to toxicology, and we had our plan was to do a critical limb ischemia phase 1A pilot feasibility trial in Ireland, and it was the first stem cell transplantation trial in Ireland. So we were dealing with a regulator who mightn't have had as much experience as other European countries, but there was an insistence that we do our toxicology using GMP manufactured products. So we had to have a GMP facility. And that it was done with the human cells, which would then require an immunosuppressed animal. So we did the toxicology. We initially did it in-house, and the regulator wanted that repeated under GLP and not in-house. So that was another issue of debate at the time. And there was no evidence of any inflammation or aberrant differentiation. The regulator also wanted a biodistribution study. So as we injected intramuscularly human cells, we developed with Cindy Coleman, who's in the audience, an assay for human DNA sequences. And we found after 3 months that the injected DNA, at least, was found at the site of injection, but not in any of the distant organs. And that's shown in that slide. In terms of marrow procurement, our regulator also put some conditions on marrow procurement, insisting that it be done in an operating theater, in a aseptic operating theater, and that the patient giving the marrow would be deemed suitable by a hematologist, and it would need to be done under a tissue procurement license. So as I mentioned, we built a GMP facility and had it licensed over a period of time, and this GMP facility was built within the university. So our university structure and our hospital structure are separate from a governance perspective, which creates some challenges also. In this initial phase 1A study, we expanded the cells in tissue culture flasks using fetal calf serum. And we decided to use an autologous approach. This study started about 5 to 6 years ago. And as it was the first study, we decided autologous rather than allogeneic. This was the batch analysis data to get the IMPD. And again, there was another engagement with the regulator at this point. These were 3 normal donors, and we used the process that we were using. But when we presented this to the regulator, they suggested that we do it in the patient subjects who we were going to enroll. Those with critical limb ischemia. We suggested we shouldn't do that, that we should do it as part of the phase 1B trial, and ultimately that was agreed. So this was a license received from our regulator for manufacture of MSCs. These are the standard IMP specification. And then the trial objectives would be to recruit the patients, do an autologous approach, and the main outcome was safety. So recruitment of patients, enrollment, bone marrow harvest, cell expansion, and because this is autologous, that takes about 2 months, or 6 weeks by the time you get quality control of released cells, and then intramuscular administration. So in the context of patient presenting with critical limb ischemia, this can also be challenging. And this is shown with this first patient who, at enrollment, he presented with rest pain. But by the time the marrow was taken and the cells were expanded, his disease, which is on your right side, his disease had progressed by the time he got the injection. The third treated patient is an example of a patient who got the cells and had a complete resolution of pain, which is a very prominent symptom in this patient. He had severe narcotic-dependent pain. After the injection of the cells, the pain resolved and the ulcers healed. Notwithstanding, this is uncontrolled, unblinded, and the possibility of placebo effects. So we enrolled 12 patients, but for a reason I'll show you in the next slide, we ended up giving cells to 4 of these patients, but there was no treatment-related adverse effects. In terms of efficacy, there was an improvement in pain in all subjects. The patient I showed you who progressed ended up requiring an amputation, but all patients had a small number, uncontrolled, as I said, unblinded, had a resolution of pain, but no improvement in acute brachial index or transcutaneous oxygen pressure. This was an observation that we didn't expect up front, that 3 subjects had karyotypic abnormalities, and 2 of them here had trisomy 5, and therefore the product was not released in any of these 3 subjects. Also, we were able to do what the regulator suggested, which is examine these cells from such patients with advanced disease. And there's a reduction in CFUF, there's a reduction in mononuclear cells, and there's a reduction in CFUF per 100,000 mononuclear cells. Also, the mean population doubling time is increased in these elderly sick patients relative to young and healthy controls. So in this phase 1B study, we used bone marrow, autologous, unsorted, flask expanded with FBS, and show that it's feasible. There's no treatment-related adverse effects, but there are karyotypic abnormalities and cell propagation problems, and also issues with patient disease progression during the autologous expansion, which would certainly suggest that autologous is going to be problematic. So I turn now to the microvascular complications. And this was an EU consortium funded under the 7th framework called Red Star. And it's designed essentially to look at the effect of stromal cells on 6 different microvascular complications across 6 different labs in Europe. So the cells were manufactured by Orbsen Therapeutics and then distributed to the 6 centers. So this cell, unlike in the first study, it's a CD362 sorted cell using a MAX system. And there's a 3,000-fold increase in purity in the CD362 sorted versus standard plastic adherent cells. The design of Red Star is shown here, where, as I said, the marrow derived cells, which are CD362 sorted, were distributed to 6 separate laboratories, one of them here in Berlin, Porto, Munich, Belfast, and Galway. And the cells were then tested in the complications in the animal preclinical models with a view to choosing one to progress to a phase 1b study. And in the interest of time, I'll just comment on 2 of these microvascular complications. One is non-healing, non-ischemic ulcers, so neuropathic, and then I'll move on to nephropathy before I finish. But in this preclinical efficacy model, we create wounds and apply collagen with cells to the wound. And looking at this bar graph, which is percentage wound closure, the group with the greatest closure were the CD362 sorted cells on Excelogen. So we move forward to do a toxicology and biodistribution study. And this is a toxicity study. Now, this clinical trial is going to be done in Copenhagen, is actually being done in Copenhagen. So our engagement was with the Danish regulator. They wanted, again, for toxicology, the final human GMP manufactured product. But we're okay with administration into a non-immunosuppressed wild type animal. So in that 10-week toxicity study, they were also okay with us doing it in our lab under non-GLP conditions. And a biodistribution study was done, which showed again that the human DNA was found only at the site of administration. So following that efficacy, the toxicology, the biodistribution, we then applied to the Danish regulator to do a phase 1b trial, which is currently underway. The bone marrow has been harvested in Galway. The cells are manufactured actually at a GMP facility in our collaborators in Leiden. And we have Excelogen, which is a collagen made in Scotland. And we've had 2 patients treated in this trial in Copenhagen. So in this non-healing ulcer paradigm, it's again using bone marrow, but now it's using allogeneic cells. The cells are max sorted by CD362 expression, and then they're flask expanded in fetal bovine serum. If I could turn to the nephropathy results. So within Red Star, the cells were sent to Munich for testing in this model of diabetic nephropathy, moving back again to preclinical efficacy. The animals have a union if they have one kidney removed, and then they develop substantial nephropathy in the other kidney, but they get an injection of either CD362 positive, negative, or plastic adherent cells, or saline alone. And again, I won't go through all the panels in detail. Suffice it to say that the cell therapy groups have an improvement in glomerular filtration rate, a reduction in proteinuria, and also an improvement in glomerular histology. So this is the preclinical efficacy data set for this diabetic nephropathy indication. So stromal cells improve kidney function and structure. And then we moved on to do biodistribution and toxicology experiments. Now we've had to do 3 toxicology experiments because one was intramuscular, one was topical, and one was intravenous. And this trial has been submitted to the Italian regulator as a consortium, which I'll describe a little bit more. And they also wanted the experiments done under GLP with GMP final product. And then we submitted that preclinical data set out of Red Star, and it was funded under Horizon 2020 to undertake the clinical trial to use a single intravenous injection of these cells in patients with declining renal function and diabetes. So that's the birth of Nefstrom, essentially, funded under Horizon 2020. And this is a follow-on from the Red Star positive preclinical results. It's a 1b/2a, so there's a dose escalation, and it's double-blind, placebo-controlled, and randomized. And it's being done in 4 centers in Europe. So actually we submitted this trial approval under the voluntary harmonized procedure through Bergamo, who were the clinical lead for the trial. We moved from flasks to a closed system, a Terumo system. 48 patients are going to be enrolled in this trial. We've done the first 16, which was the low dose cohort. And as I said, it's funded by the European Union. This is the Terumo system with reduced operator steps and reduced microbial contamination, the reduced cost of goods. We also did the expansion on this occasion in human platelet lysate. So we moved on away from fetal bovine serum to human platelet lysate. One of the challenges with this system is it needs to be coated with cryoprecipitate, and we have a difficulty getting access to cryoprecipitate. We have a team of GMP facilities, including our one in Galway, Leiden, the UK, and Bergamo, who are making the product. And the design essentially is that the first CD362 sort happens in Leiden, and then Leiden distribute the cells to the other 3 GMP facilities where the cells are manufactured. And this is the clinical trial description. As I said, it's just finished its low dose cohort, and it's enrolling patients who've had 3 years of declining renal function at a specific rate. So in this study, we're using bone marrow. It's allogeneic, max sorted based on CD362 expression, it's bioreactor expanded, and non-xenogenic using human platelet lysate. So I've gone through our preclinical efficacy and toxicology studies. I made some reference, as I said, to the issues of preclinical models, and are they good predictors of efficacy in humans, or are they just meeting a regulatory requirement to show some evidence of rationale in animal models? I talked about toxicology and how our discussions in relation to toxicology varied across some of the different regulators in Europe. I talked about moving from fetal bovine serum to non-xenogenic, and some of the other challenges I would allude to include excuse me, maintaining a GMP facility in an academic center. So that's a significant cost. And also having the university function as a sponsor for first-in-man human trials. So all of these are issues that I think will be faced by a number of different people in this field. So we've been considering the source and procurement of the MSCs. All the work that I presented was bone marrow, but we're working on umbilical cord also, whether it should be autologous or allogeneic, sort or bulk manufacture, the method of the sort, MAX. We've also got ongoing work with FACS, homogeneity of the starting material, flasks versus bioreactor expansion, passage and population doubling, xeno-free manufacture, cost of goods, automation. And Melissa Van Pell, who's from Leiden, who presented yesterday, showed that even when you control for all of these things and you take a marrow and split it in 3 across 3 sites adhering to the same SOPs, there will still be product variability between the 3 sites. So ultimately, we'd like a therapeutic product with umbilical cord, allogeneic off the shelf, GMP compliant, FACS sorted, bioreactor expanded, xeno-free with robotics and inline monitoring. This is our GMP and our clinical research facility and our tissue establishment group. So I'd like to thank them for their participation in these. These are some colleagues of mine, many in the audience, like Frank Barry and Mary Murphy, who've worked on mesenchymal stromal cells for a long time, Linda Howard and Cindy Coleman. And these are the funders. Most of this was funded by the European Union, but the critical limb ischemia trial was funded by Science Foundation Ireland and the Health Research Board of Ireland. Thank you.
Thank you very much. Are there any questions? I think we have time for one quick question.
So, over here.
Yeah.
So how is your reproducibility in the allogeneic product? That is to say, when you have different patients or different donors, Is there always the same activity, or do you have to sort that out? And following that, would that maybe mean that at some point one should use iPS cells, for example, to do MSCs, to produce MSCs from iPS cells, where you have a more a homogeneous product then in the end of the day? What is your thought on that?
There is a degree of heterogeneity with allogeneic donors, and again, you would have seen from Floristem today, they have potency assays. We're at phase 1B and 2, so we're developing potency assays, but it will be necessary to do potency. I'm not sure, I don't know whether iPS-derived MSCs would have a difference in terms of donor-to-donor heterogeneity, but it certainly exists in the allogeneic bone marrow-derived context.
I have 2 short questions. First of all, you use the IV approach for your treatment strategy, and it is well known that the MSCs stick in the lung. Do you have any other experience to, let's say, convince us that the clinical results are really coming from the MSCs, and how long they are circulated? This is one point. And the second point is, it is, let's say, questionable at which stage the cells can impact the renal function. If you have a glomerulus which is fully sclerosized, then it is hardly to believe that you can reverse this end-stage disease. Also, which patient you are include from the histological features, and what you expect beside of clinical function?
So in our administration paradigm, is topical for non-healing ulcers, intramuscular for critical limb ischemia, and intravenous for diabetic nephropathy, as you point out. And we've done biodistribution studies after the intravenous injection, and like you mentioned, a lot of them go to the lungs. But actually, using cryovis imaging, there is a redistribution to the kidney, the remaining kidney. There's only one kidney in these urinonephrectin mice animals, and that dissipates over about 5 to 7 days. So there is some residence within the kidney after IV delivery. The second question about sclerosis, we're including people between GFR 30 and 50. We don't do biopsies. There's no biopsy in the protocol. In the animal model, we would have injected in advance of advanced sclerosis occurring in the majority of glomeruli. So I think if you have a GFR of 50 down to 30, and they would have to have had a decline in the preceding 2 to 3 years, we believe there is an opportunity to recover something. But you're absolutely right that if there's a sclerosed, fibrosed glomerulus, that's not going to become podocyte-rich and filtering anytime soon. So it's probably aiming to protect those glomeruli which are partially damaged. Thank you.
Further questions? Okay, thank you so much, Timothy. And now we come to the last talk, which is given by Michela Gabaldo from Fondazione Teleton. And she speaks about an integrated ecosystem of research and development for orphan medicinal product, and about development of a guidebook, which is of importance. Thanks.
Thanks. I'm Michela Gabaldo. I'm here representing IRDIRC, the International Consortium for Rare Disease Research, and I'm here as a member of a therapy scientific committee who have developed this orphan drug development guidebook. I'm also head of alliance management and regulatory affairs at Teleton Foundation. So this is the current rare disease landscape where we have more than 400 million people affected by rare disease. The majority of them are children, and approximately 80% of the rare disease are of genetic origin. What is impressive here is that only 5%, less than 5% of the rare diseases have an approved therapy at the moment on the market. So, of course, this is highlighting the fact that there is a tremendous unmet medical need, and the IRDIC goal, and this is the reason for why IRDIC proposed a new goal that is quite challenging, of developing new rare disease treatment, 1,000 new rare disease treatment by 2027. But however, with all the incentives that has been put on the market in Europe, in US, and in Japan over the last year to accelerate and help the development of rare disease drug development, the current rate of drug development is approximately 40 new therapies per year. So with this rate, of course, will take centuries to reach that goal. The other situation is that this is the current development process for a drug for a large indication, where there is a consolidated path, well known, based on risk management and gate review, where there is almost linear progressive increase in the cost and the long-term predictability. Of course, this is the standard one. In the case of the rare disease and the rare disease orphan, ATMP, of course, development, this is even more challenged because most of the time we have a challenge in having the animal models to develop the drug. We have challenge in completing the clinical package leading to the registration. So we have not complete pharmacological safety and efficacy package. And of course, if you add these to the reimbursement challenge, a sustainability challenge, this is why that this current development process is not applicable. So what we have seen over the last year is that the regulatory agency have tried, and also the central bodies have tried to put in place and provide initiative and resources to accelerate the drug development for rare disease, to accelerate and enable the development of innovative and potentially transformative drugs. However, at the moment, the impression is that all the developers are having a sort of disorganized toolbox. So it's not, and what is missed is something that is well organized and standardized, so that a sort of organized toolbox that can be used by any developer. In particular, all the striking technology that are arriving on the market are developed by academia and small medium enterprise. So this is a need that we see even more important. So starting from this, the Therapeutic Scientific Committee have decided to launch the Orphan Drug Development Guidebook project, where we have been with the aim to describe all the available tools, incentives, resources, and practices specific for the development of traditional, but also for the innovative drugs and therapies for rare disease. And we have mapped out how to use them. So we have engaged 27 drug developer experts coming from industry, coming from academia, coming from patient organization, and coming from regulatory body at the central level, at the national level, across the three geographic areas, so US, Europe, and Japan. And we have with them defined which are the framework, so divided by the different phases and defining the milestone along this drug development pathway. We have identified what we call the building blocks. So we have identified those tools, initiative, resources, and practices that are used during the drug development. And for each of them, we have created a sort of fact sheet. And we have mapped out 3 case scenarios that I'm going to explain you in a second, defining at the end a roadmap with a checklist of what to do and when to do it. So these are the questions that has been posed to the task force. So which are the essential steps through the development of an orphan drug? What are the important committee tools, incentives, and what is the most efficient way to use this available initiative? Which are the pros and cons of the different initiatives, and where are the gaps and the potential needs for new initiative? And we have mapped out 116 building blocks, and we have grouped them in 5 clusters. So the first one is regulatory, so those pathway designation incentives for orphan drug development in Europe, US, and Japan. Then we have selected those HTA reimbursement initiative that has to be taken into account very early in the development so that you're able to overcome or minimize the burden at the market access level. We have mapped out the early access initiatives, so programs that enable patient treatment before the regulatory license at the central level or the local level that may be provided reimbursed or provided at no cost, according to the local regulation. And then, of course, we have mapped out the development practice, so best practice established by the developer in the field of rare disease to improve orphan drug development in terms of speed, quality, and efficiency, and also resources. And analyzing these as expected, we have found that more than 50% were fully under the regulatory cluster. There was a minority on the HTE, but not because they are only few building blocks, but just because we selected those that were relevant to be considered during the development. There was a minority part for the early access, and the remaining part was the development resources. And we ask, this is the standard development framework where you have the patient need, you define the disease knowledge, you define your target organ, and then you start all the development path. So what we ended up was to clarify that this development path and this guidebook that they were developing was not suitable for the development of medical device, but at the same time, it was too complicated to consider also the repurposing and the line extension. So we took out from our development path, and also the last part after the registration leading to the access, we combine in a single market access building block, so that we could focus on starting from the patient need and arriving to the registration, to the market access, and then responding to the patient need. And we put in a linear way the development path, and we identified for these key milestones, which were the milestone key for the progression of the program, and define which tools we have to use before and after. So we, to maximize the clarity of this guidebook, we have also defined in 3 categories the product, under development. So we started from the standard orphan drug, where you have a traditional, well-understood pharmaceutical product like the small molecule or like the well-characterized protein biology, like enzyme replace therapy or antibodies, where you have a non-negligible body of knowledge around the disease, both in the regulatory field and the medical community, where the patient population is sufficiently understood and there are animal models possible, and the disease is present both in children and old adults. And then we ended up in defining other 2 more challenging categories. The first one was the less understood, what we call the less understood technology, that is advanced therapy in principle, and then less understood disease. In terms of less understood technology as compared to the standard traditional small molecule or well-characterized biologic, of course, here we have gene therapy, stem cell therapy, and gene editing, where in this case, in this clustering, again, the challenge was not on the disease because there were enough body of knowledge on the disease, and also the patient population was sufficiently known, and the disease was present in both children and adults. And we arrived to the far front, the less understood disease, where there is a very low prevalence, where there are a negligible body of knowledge on the disease, where natural history study is little known, and there are no, the disease exists only in a pediatric population, there are no biomarker or clinical relevant endpoint. And these are the case, if you combine the last 2, this is the case of the majority of the orphan drug development, orphan ATMP that has been developed as per today. So we ask when you start developing a product, a rare disease one, we thought that it was important to start from what? From the stakeholder mapping, so mapping who are the stakeholders that has to be involved during the development, map which are the information that are available already, define which financial resource you will use, and then define the target patient value profile. So in a snapshot, this is sorry, there is a problem with animation, but so in terms of stakeholder mapping, try to see whether there are already patient organization and the community advisory board already available for that disease, like, for example, the CAPS or Eurordis. Try to see whether in your geographic area there are stakeholder networks specific on that specific ultra-rare disease, like, for example, in Europe, the ERN, in the US, the CRN, in the Japan, the AME, the ERUT, whether there are general development support platform and infrastructure like the C4C or the AGP. And then define and map which are the available information on the disease in terms of whether there are national history study, diagnostic tool, patient clinical outcome measure, and whether there are biomarkers already available, and whether your rare disease has been already coded. Of course, define if you are using, if you can use public funding or private funding, and then define the target patient value profile. So what is important here is that as soon as possible, regardless of where you are starting with your development plan, you have to start from the start and mapping all the knowledge that you are available. And in case something is not available as a result of this gap analysis, you have to fulfill immediately. So due to the time limited, I'm going here to show you only the snapshot about the regulatory and access building blocks and how we put in place. So this is the development pathway, and here are the key milestones, the main milestone with a dotted line that we have put in place. And for each of them, for example, starting from the orphan drug designation, that is the simple one, across Europe, US, and Japan, we have built 3 fact sheets for each product. But then in this roadmap, we have identified also with a double-headed arrow what is the time frame when you can apply for this orphan drug designation. So the minimum you need to have completed and have the data from your clinical proof of principle, but then you can ask until before the NDA BLA submission. But then also with the dotted, with the empty circle, we have identified also what is the best time, in our view, based on the expert experience, to apply for this. And in the fact sheet, we have clarified why this is the best time to maximize all the incentives for this. Then we have mapped out, of course, whether there is a possibility of early interaction. And with the color code, you have those that are in gray are the standard orphan drugs. So this means that those in gray are available for any kind of the 3 category product, whereas the red one are available only in this case for advanced therapy. And then whether you may have interaction with the European joint EMA FDA or US only interaction. Here you have the EMA protocol assistance, and again, what is the best time, what is the time frame when you can apply, and what is the best time at which stage you have to, it's better to apply. One important thing that is more relevant for Europe is the scientific advice at the national level with the member state, and in particular the importance to maximize the interaction with them before starting the first clinical trial to maximize then the possibility to enter the trial. And these are all the other about FDA. And what is important is that in this guidebook, we have also highlighted with a dotted row and with the empty circle when it is important to start thinking about, for example, of the parallel EMA FDA scientific advice. It's important to start well in advance to think about this, but then apply later on in the development. And this is the Sakigake initiative for Japan. Those in red at the moment here are specific only for ATMP, so the classification or the certification, or the regenerative medicine advanced therapy classification with FDA. And then we have also EMA qualification initiative that are available for when you the product is falling with under the what we call disregarded disease area. And in the US, of course, we have also the FDA qualification. So Then we have mapped out the regulatory accelerated program, and again, those in gray that are applicable to any kind of product, and then those that are eventually available only for ATMP. And then we have mapped out the initiative for the patient market access. I'm not going to the details of this, but what is important to highlight is that for each of them, there will be, at the moment, the guidebook is static, but we are planning to put it interactive so that as soon as you click on the name of the initiative, then it is open up a pop-up with all the fact sheet describing the initiative and all the pros and cons of this initiative. So I'm going to conclude, and what is important is that this task force have highlighted the urgent need to overcome the lack of standardization in the rare disease drug development. And what is important is to start not from idea, but start with from the patient need. Wherever you are starting your development plan, make the start, so to assess the level of understanding of the disease, mapping your stakeholder, interact as soon as possible with early in the development to seek regulatory advice, consider early on the strategy for eventually apply to accelerated regulatory approval, and keep in mind the early interaction also with the HTA bodies and payers, which is very important. And so, in other words, and with the patient need rather with the patient care in mind. So we think this guide will be available on the ERDIRC website. We are finalizing and we are launching in the next few months, and will be available to any developer. So, and we are expecting that it will be of great use for academia and small medium enterprise. So these are the 28 experts that have built these guidebooks, and this is their affiliation. Thanks a lot.
Thank you very much for your talk, and I think everybody can underline your statement that we have to focus on the medical need rather than other things. But based on the time, please contact Michela in the lunchtime for discussing issues which can help to pushing up the strategy in advanced therapies, not only for orphan drugs, because this is, let's say, usable for all other strategies in advanced therapies. Again, we as the chairs here have to thank everybody for participating on this session, for the discussions and listening, and have a good lunchtime. Thanks.
Del primo prossimo. Sì.